Hi everyone. So it's great to be here, and thanks everyone for coming. So I'm a PhD student in Finlay's lab, and in our lab, like as a lot of labs today, we use genome editing techniques to understand variants effect at scale. And my project focuses on rare human variants, aiming to determine which one are disease causing. So rare diseases are rare, but collectively they are common, and they affect like more than 350 million people worldwide, spreading across 7,000 diseases, and 80% of those have a genetic origin. So the development of next generation sequencing has brought like opportunities for diagnosis, and large whole genome sequencing projects have been developed, such as Genomic England, that have recruited over 70,000 participants for the rare disease program across like 190 conditions. So they have collected whole genome sequencing data and along with medical record. And now it's more common to get patients' whole genome sequence, which has the potential to improve diagnosis. But unfortunately, still 50 to 70% of rare disease patients remain undiagnosed after having their whole genome sequenced. And that means that a lot of families are left without answers, like which delays treatment, reduce life expectancy, prevent them from participation in clinical trials, and add emotional and financial burden. So to summarize, like many patients with rare disease lack a genetic diagnosis, and this is largely due to the lack of understanding of a functional effect of variants. So as we know in this conference, there is like 3 big ways to look into variance effect. And it's like computational predictors, functional assay, and genetic analysis of cohort. And in my project, we bring these 3 strategies into one single framework, aiming to identify as many pathogenic variants as possible across the whole genome. So the framework consists in 3 simple steps. So the first one is aim to reduce the number of variants by prioritizing the best candidate variants, so the variant that could possibly be deleterious. And then we want to test all these prioritized variants using genome editing in human cell line. And finally, we want to validate the novel loss of function variants revealed by the genome editing screens by linking the variance effect to the carrier phenotype. So to start with the prioritization, so we want to select relevant regions in the genome, and we select genes that are associated with disease and are also essential in our cell line. And we end up with 143 genes, and then we go in Genomics England, whole genome sequencing data, and we retrieve variants within this gene and also upstream and downstream regions. And then we want to keep variants that are predicted to be pathogenic, and for that we use CAT and SpliceAI with a score above 25 and 0.2. And finally, we want the variants to be efficiently installed in the cells to realistically test for loss of function effect in cell culture. So we end up with this test set of Genomics England variants that are majorly composed by missense variants, and most of all the variants have an unknown effect. And to this, we add some control variants, so common variants from UK Biobank, which is a non-disease specific cohort. And then we add some synthetic nonsense protein truncating variants, and finally variants that are pathogenic and likely pathogenic in ClinVar, which is a public database of genetic variants that are reported with their clinical significance. So here, what we really want to ask is like, which of these 13,000 variants from Genomics England are driving phenotypes? And to answer it, we use prime editing. So Michael and Christina from my lab, they have developed this platform that allows in one single experiment to test any variants anywhere in the genome at scale. And for that, they use prime editing, and prime editing is a precise search and replace genome editing methods that use a Cas9 nickase fused with a reverse transcriptase, and is guided by a specialist RNA sequence that's going to introduce a specific DNA change at the targeted site. So Primating allows us to insert any short whole genome sequencing variants, and most importantly for us, one synthesized oligo will give us like one pegRNA, which will allow us to have like one edit in the genome. And therefore it's cheap, and it can be applied to many genes genome-wide. So we use primating in a framework very similar to CRISPR-Screen. So in my experience, I started by designing over 20,000 pegRNA, each encoding for one specific variant. And then a lentiviral plasmid library is generated from a pool of oligonucleotides. And on day 0, we transduce the cell with the library, and we obtain a cell library. And then the edit will take place. And what we do next is like we monitor the change over time by performing the deep sequencing of the survival cells at 4 time points. And the experiment was performed with 2 replicates. And when we deep sequence the cells, actually we only sequence the pegRNA construct. And that allows us to measure 2 things. So the first one is like the pegRNA abundance over time. So by counting the number of reads for one pegRNA at each time point, we can track his abundance over time. And a strong depletion of one specific variance, a strong depletion of a specific pegRNA suggests that the variant it introduces is likely to be loss of function. So the trick here is like together with the pegRNA in the pegRNA construct, we also add a surrogate target, which is a sequence that mimics the genomic site. And that allows us to, by measuring like how many reads the surrogate target contains, the intended edit, we can quantify how efficiently the pegRNA introduces the variant. And that's like a high throughput system to evaluate the pegRNA performance. So for here we have like and we have seen many plots of those plots today, but this is the pegRNA fold change from day 20 over day 4, and it reflects the variant's impact on cell survival. So where greater depletion suggests loss of function, and this is consistent across replicates. And we use nonsense variants as positive control, which should deplete over time if they are successfully inserted, and common variants as benign variants that are negative control and should remain stable. And it's encouraging that we see like some nonsense variants that are dropping more than common UK biobank variants. But here I'm just showing like the fold change between 2 time points, but in reality we have way more data. We have 4 time points, 2 replicates, and also the pegRNA editing efficiency. So to fully capture the functional effect of a variance, we need a composite score that integrates all this information. And we did this by training a random forest classifier model using only experimental features. And we train it on like control variants. And if we plot so we successfully like scored 11,404 variants. And when we plot the genomic segment variants and color them by our new loss of function score, we can see that variants that have like are successfully installed with high primating efficiency and are like greatly depleted by day 20, they tend to score highly loss of function, as we expect. So if we look at the model features, and the most important one that are like on the top, we get all the pegRNA depletion features, and then followed by the variance editing efficiency. And the cleanest time point and replicates tend to be on top. So then we want to look like how the model perform on genomics England data, and so that's the number of genomics England variants, and we put the variants into loss of function score bins, and we can see that very few variants are scoring highly loss of function. And here I'm going to just change the y-axis from counts to proportion to talk about like the ClinVar annotation of these variants. So first of all, like most variants are not in ClinVar, and we have a lot of variants that are like conflicting or uncertain. But we can appreciate that like the predicted probability to be loss of function in our assay is higher for all the pathogenic and likely pathogenic variants in ClinVar, and the benign and likely benign variants tend to be in the lower bins. So if we zoom into the two top bins, we have 492 genomic singlet variants, 440 have unknown or unclear significance in ClinVar, and from the all 436 likely benign variants in ClinVar, we have 2 that are within this top bin, so potentially false positive. So next, we wanted to compare these bins to predictors, and here we have alpha missense, which predicts missense variants, and we have all our missense variants, and we can see that there is a correlation between the two metrics. And we did the same for our splice variants using splice AI, which predict how genetic variants affect RNA splicing. So now we are currently investigating this high-scoring loss-of-function set on the top bins to identify the ones that are underlying diseases. So for that, we can go back to the participant that is carrying a specific variant and look into, is the variance located into a gene that is associated with the carrier phenotype? What's the disease mode of inheritance? Is the variance a de novo? Or is the variance seen in other cohorts, such as like UK biobank in healthy individuals? And we have some variants, such as this missense variant, and it ticks all the boxes. And actually, this variant has been previously identified as a genetic cause of the participant rare disease phenotype. But we have other variants, such as this one, that is like carried by someone that doesn't have a diagnosis. So to summarize, like the vast majority of new loss-of-function variants that are found in carrier, we are still undiagnosed, and so far in the essay, we have only 9 variants that are in patients that have been solved that had a genetic diagnosis. So today I presented a framework that successfully identified new loss-of-function variants from whole genome sequencing data across 101 gene regions, providing, by testing like over 11,000 prioritized variants from genomics England and providing 492 new functional hits. And it's guaranteed with many false negative and incidental findings, but it has the potential to expand the frameworks to new disease association. And with that, I would like, because this project does not only reflect like a lot of data, but also the support of people around me, I would like to thank my amazing lab, as well as Greg for the exceptional support and guidance, my TC committee for the precious scientific insight, the Quick platform, and very importantly, the 100,000 patients for their trust. Yeah. And I will take any questions.